a presentation entitled, a paper entitled Mirroring Liver Repair via Transient Activation of Regenerative Pathways in Hepatocytes Using Lipid Nanoparticle Complex Nucleoside Modified mRNA. Try saying that 5 times fast. All right, take it away, Rakshith.
Thank you. Good morning to everyone. Today I'm first year PhD student, and I work at Dr. Mawangi's lab. I'm Rakshith Kumar. I'm sorry. So today I'm going to present an article from Nature Communications Journal titled Murine Liver Repair via Transient Activation of Regenerative Pathways in Hepatocytes using Lipid Nanoparticle Complex Nucleoside Modified mRNA. This article has got published in January 2021. I chose this article because I am currently working on nucleoside modified mRNA for vaccine development, and I'm interested to learn more about the mRNA based therapies as well, because it's another application of use of mRNAs, and it's very popular these days because of the development of vaccine for COVID-19. And this image is the liver, which is affected with the steatosis, which is a metabolic disorder, and this is a histopathology picture, and this is a histopathology of the steatosis. And this is a mRNA, which is complexed with a lipid nanoparticle, and these are the mRNA, and these are lipid nanoparticle. To begin
We're
with,
seeing the presenter view right now. Just so you know, we're not seeing the presentation view. There we go. Perfect. Thank you.
now can you see the presenter view?
Now it's the presenter view.
Okay, you can't see it.
That's the presentation mode.
To begin with, first I will give the introduction on liver and its functions very briefly, and then I will talk about fatty liver disease. Then I will talk about hepatocyte mitogens, which are used in this paper, hepatocyte growth factor and epidermal growth factor. And I will discuss how they have used the mRNA, which is encoded for hepatocyte growth factor and epidermal growth factor in liver injury models. And then I will talk about the mRNA therapy, a new approach to medicine, and in vitro mRNA synthesis, how we can synthesize in the lab. And later I will shift to the paper. I will talk about the objectives, methods, results, and I will summarize the paper, the results. And then I will talk about the pros and cons of the paper, and then I will end the presentation with what we can do in future with the use of these results. Liver, it is the second largest organ in the body. It is located at the upper right abdomen under the ribs, beneath the diaphragm and top of the stomach. In case of humans, but in case of mice, it is present in the ventral part of the body. It is covering almost all peritoneal cavity in case of mice. And the liver consists of 4 lobes, mainly the median lobe, caudate lobe, left lobe, as well as the right lobe. And this is anatomy, and for development of models, few of the lobes like the left lobe and the caudate lobe will be dissected to make partial hepatectomy. And the major function of the liver is it converts nutrients into substances that body can use. For example, carbohydrates, proteins, and fats get metabolized into their simpler forms to make use of the energy. And also, if there is more sugar in blood or the glucose content, it will be converted into glycogen and store in the liver. And also, liver is a storehouse of various vitamins and minerals, and it also filters blood, detoxifies the chemicals, and metabolize the drugs into reduced forms to make them non-toxic. Fatty liver disease are also called as steatosis, means the accumulation of lipids in the liver due to any metabolic changes like increase in the glucose level or increase in the lipid level in the blood. So all the fat will get stored inside the liver, and when it becomes excess, it will cause steatosis. It is the most common chronic liver disease in this modern world because of the change in the lifestyle. And in fatty liver disease, there are two main types. One is alcohol-related fatty liver disease and non-alcoholic fatty liver disease. Alcohol-related is because of chronic alcohol intake, and non-alcoholic fatty liver disease is having many, like it's not having a specific etiology. The main risk factors of non-alcoholic fatty liver disease are diabetes, obesity, and other metabolic diseases. It is having a global prevalence of 25.2%, and it's very important to mitigate this. And once we get NAFL, the sequela will be uh, once the inflammation begins, it will become steatohepatitis, and it may progress into liver cirrhosis in the chronic form, and then it may cause the liver failure and death of the individual. Gross and histopathological changes in steatohepatitis, which is very important to know because to do the animal trials to test the drugs and all. So this is a gross morphology of the liver in the normal state. The liver color will be dark brownish red, but when it is having with the steatosis, it will be yellowish to discolored color. Discoloration of the liver will be there. And also in HND staining, this is a normal architecture of the liver, and this is a steatotic liver with the vacuoles present inside the hepatocytes, which depicting the lipids stored lipid inside the hepatocytes. And this is oil red rose staining, which is commonly used to stain the fats. So this is the normal, and this is a steatotic liver where we can see the red droplets. Each droplet represents the lipid droplets inside the liver. And so this is also HND staining, which is showing the macro size, means more amount of fat accumulation inside the hepatocytes. And this give a very popular pathognomonic lesion like signet ring appearance, means the fat will push the nucleus to the periphery and it look like a ring. Each cell represents one hepatocyte, and this is the nucleus. Hepatocyte mitogens are the proteins which will uh, help in the proliferation of the hepatocytes. In this article, they have used HGF and EGF inhibitor, that is, they have used epidermal growth factor. These both can be used in making the liver better, means to reduce the hepatocyte damage or liver injury. How the hepatocyte growth factor will function is it reduces the abnormal level of cholesterol and other other lipids, and then it induces the antioxidant response in hepatic cells by increasing the glutathione and related enzymes like GSH peroxidase and GSH transferase, and also it decreases the reactive oxygen species, so it mitigates the liver injury. And epidermal growth factor helps in the regenerative response of the liver through inhibition of the intrahepatic lipid accumulation. So the steatosis can be mitigated by using these two. And there are papers in and also other drug platforms or the protein therapies where the hepatocyte growth factor and epidermal growth factor both are given together and they perform synergistically to promote the liver regeneration by increased cell proliferation and metabolism. And in this article, for to develop chronic liver injury model, they have used CDE diet. What is CDE diet means? It is choline deficient ethionine supplemented diet. Uh, how does choline deficiency will cause a steatotic model is choline, if it is deficient, it cannot synthesize, means phosphatidylcholine choline cannot be found in the body. And phosphatidylcholine is an important component in VLDL, means very low density lipoproteins or any other lipoproteins. If the lipoproteins have not formed, they cannot transport the lipids from the liver to other tissues. If the lipids from the liver is not transported to other tissues, the lipid will be stored inside the liver itself to cause the steatosis. And also phosphatidylcholine is an important component in the formation of cell membrane also. In the absence of phosphatidylcholine, there will be cell dysfunction and structural damage to the cells. And also by giving the choline deficient chow and also ethionine supplemented. Ethionine is supplemented in the water at 0.1 percentage, and it is a competitive analog of methionine, and it will disrupt the protein synthesis in hepatocytes. In by giving combination of both of these food in the food, both of these combination, it will cause chronic liver injury model in mice by accumulating the lipids in the hepatocytes, inflaming the liver and causing hepatic necrosis. After the necrosis, the alanine aminotransferase, that is ALT levels, will get increased, and it is a liver specific enzyme which is actually used in the liver function test to know if the liver is healthy or not. After chronic liver injury model, they have used acute liver injury model, and they have used overdosing of acetaminophen, which is abbreviated as APAP, uh, to develop acute liver injury model. The mechanism of causing the liver injury is acetaminophen is metabolized in liver, and after metabolized, so it will uh, give a toxic metabolite as N-acetyl P-benzoquinone, that is NAPQI, and this NAPQI get reduced by antioxidant glutathione into a reduced non-toxic form. In case of overdosing of the acetaminophen, this GSH will get depleted by reducing more and more, and then it will get completely depleted, and this NAPQI will take the upper hand, and then it binds the mitochondrial proteins in hepatocytes to cause the necrosis and then to cause acute liver injury model. And to mitigate these two models or the liver injuries, they have used mRNA therapy. mRNA-based therapy is a new approach to medicine. It is based on triggering the synthesis of proteins within the cells. mRNA, mRNA will be having set of instructions to make proteins inside the cell and send them to different parts of the body. And this technology is highly potent, can be rapidly developed soon because there is no need of any propagation of cells, vectors, and any other things. And it is non-integrative and it is safe because this won't enter into the nucleus, and also it is safe because there is no involvement of any viral vectors or any other um, like attenuated viruses like that. And it is easily scalable and low cost production. uh, and recently, as I said, mRNA based vaccine got approved to treat COVID-19, and also there are many preclinical studies going on for vaccine development in rabies, Zika, influenza, and HIV infections. Uh, what were the limitations of mRNA in therapy previously were mRNA is very unstable and get easily degraded by nucleases, and also it activates the immune systems, toll-like receptor 3, 7, and 8 when we administer them. And also it is having high negative charge density, and it reduces the permeation of mRNA across cellular membranes. To mitigate this problem, so nucleoside modified mRNAs have been introduced and discovered very recently. These nucleoside modified mRNA or mod RNA are some of the nucleosides from the classical mRNA are replaced by naturally modified nucleosides or synthetic nucleoside analogs, like uridine is replaced by N1-methyl pseudouridine, and cytosine is replaced by 5-methyl cytosine. And by replacing these nucleosides, this makes mRNA more stable and to evade innate immune systems and also have high translatability. I will briefly tell like how uh, mRNA can be synthesized in vitro. So uh, mm, a plasmid which is containing an open reading frame of a gene of interest, which also have 5' and 3' untranslated regions, and also a T7 promoter can be used, and also it should have a restriction site downstream to the insert gene. And then by using restriction enzyme, we can cut at one site and then to make a circular plasmid DNA into a linearized plasmid DNA. And then by using PCR, we can introduce a poly T tail, because our mRNA should contain a poly A tail to have more stability and more translatability. So we can introduce a poly T tail, and then after using this template for in vitro transcription reaction, this can give us a mRNA with a poly A tail, and also we can co-transcriptionally or enzymatically we can add a cap to increase its stability as well as translatability. And also there are different kinds of caps nowadays, like modified caps, which are synthetic caps. So this is procedure. And how to deliver mRNA. So we can deliver mRNA into the cells with micro injection technique, RNA patches, gene gun, protein condensation, RNA adjuvants, and also by complexing with lipid nanoparticles. In this article, they have used lipid nanoparticle complexing to deliver the mRNA into mice. So coming to the article, objectives of this article are to determine the organ specificity and cell specificity of mRNA lipid nanoparticle which is injected by intravenous route, and also to evaluate the efficiency of nucleoside modified hepatocyte growth factor encoding mRNA on hepatocyte proliferation, means how this mRNA will perform with hepatocyte proliferation once we inject through the intravenous route. Also, they studied this mRNA, which is encoding for hepatocyte growth factor and epidermal growth factor in chronic injury models in CD that induced chronic liver injury model, as I explained before. And also they tested the same mRNA combination of HGF and EGF in acetaminophen induced acute liver injury model. mRNA production in this article, so they have the same coding sequence, 5' and 3' UTR, 5' cap 1 structure. Cap 1 structure means it will be, it's chemically it is 1-methyl pseudo 2' O methyl transferase cap 1, and also it have poly A tail, and it is encapsulated in a lipid nanoparticle. And this coding sequence, so they have used multiple mRNAs for control, treatment, and also treatment will be the HGF and EGF encoded mRNA. For controls, they have used firefly luciferase encoding coding strand, and also enhanced green fluorescent protein encoding coding sequence. These two are for controls, and also they have used policy RNA as a control in some of the studies. And this mRNA is produced by T7 RNA polymerase kit using, and also this mRNA is containing 101 nucleotide long poly A tail, and as I said, it is a nucleoside modified mRNA. So they have used 1-methyl pseudouridine 5' triphosphate instead of uridine triphosphate. Methods and results. So I'll tell parallelly both. First I'll tell the methods and the results of them. Study 1, evaluating two different intravenous injection routes and duration of protein expression by lipid nanoparticle complex mRNA. They, in this study, so they evaluated the organ distribution and also duration of protein activity on intravenous injection of firefly luciferase encoding mRNA. They they choose two routes. One is tail vein route as well as retroorbital sinus route. So they injected firefly luciferase encoding mRNA on tail vein injection as well as retroorbital sinus injection at the dose rate of 5 microgram per 20 gram of body weight of mice. And the protein expression was studied by measuring the bioluminescence detected on the luciferin and the luciferase reaction on IV spectrum imaging system. So there will be a production of the bioluminescence once the luciferin and luciferase both get reacted. And the bioluminescence is measured at 5-hour time point, day 1, day 2, day 3, and day 6, day 8 time points. And these are the results. On tail vein is showing the upper row and the retroorbital sinus is showing the bottom row. Tail vein and retroorbital roots both showed the bioluminescence on day 1 as well as in 5-hour time point in case of tail vein. And it gradually reduced on day 1, day 2, day 3, day 6, and finally I come to baseline as you see in the graph, baseline on day 8. And so they evaluated the duration of the activity of the mRNA injected through the intravenous route. And they also confirmed that the mRNA which is injected through the intravenous route will reach the peritoneal cavity into the liver. But this is because the mRNA LNP which is injected through the intravenous route will bind to the apolipoprotein E, which is present in the blood, and that in turn targets the AP receptors in the liver. And also they concluded either of the approaches can be used for transient protein expression in liver. Uh, but in this article, they chose the tail vein route because this is very easy and it is not. It's a human practice. Like retroorbital route is very difficult to perform in mice. Every mice. After confirming that the mRNA will reach us to the liver and it gives the transient protein expression for 8 days, so they evaluated the histologic specificity of intravenously injected mRNA LNP. So they checked the organ affinity for intravenous injected mRNA complex with lipid nanoparticle. Here they used EGFP encoding mRNA at a dose rate of 10 microgram for 20 gram mice, and also they used policy mRNA as control. And 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 after the injection, they left for 5 hours, and then they sacrificed the mice, and then they collected the liver, lung, spleen, and intestine samples. They did the cryosectioning procedure for getting the cryosections of these organs, and then they performed the immunostaining. Uh, the poly-C RNA didn't. So this is the section of liver. The poly-C RNA did not show any fluorescence because it didn't had the protein. Uh, but the mice which were injected with EGFP encoded mRNA showed the fluorescence in the parenchyma of the liver. You can see here the green fluorescence after the 5 hours time interval. And also they did the staining, same staining for in case of spleen tissue, lung tissue, and intestine tissue. In spleen, the parenchymatous and all the policies were negative because it didn't contain fluorescence, and the lipid nanoparticle is always negative. Um, but the spleen didn't show any fluorescence in the parenchymatous region, even the lung or the intestine. But in spleen, only the red pulp region was showing the green fluorescence. This is, and also they did the co-immunostaining with CD45 and CD31. And then they confirmed that the cells which were giving the fluorescence were not the parenchyma cells, they were the blood and the endothelial cells which were present in the red pulp region. And also in lungs, the fluorescence were very distributed all along the tissues, and they did co-staining with CD31 and CD45. So they found that they suggested that they were the alveolar macrophage cells because they were positive for CD45. And in intestine, the lamina propria, the, the lamina propria region was only positive, and they confirmed that they are small blood vessels and lymph vessels and blood cells by doing co-immunostaining. Hence, they concluded that the liver is the main target organ for lipid nanoparticle complex mRNA, which encodes for EGFP, injected through the intravenous route. But still, it transferred to some population of the endothelial cells, hematopoietic cells in spleen, lungs, and intestine. After confirming that the mRNA is transfecting into the hepatocytes and translating the proteins in the liver, they wanted to confirm that which cells in the liver are actually getting transfected and producing the proteins, giving the fluorescence. So they formed two groups. One, they gave the control injection, which has poly C mRNA with the lipid nanoparticle, and the other group was injected with EGF protein, enhanced green fluorescent protein, mRNA, which contain the lipid nanoparticle through the intravenous routes only. And then they stained for EGFP. Along with that, they co-immunostained with HNF4 alpha, which is a liver cell-specific marker for hepatocytes, and CK19 marker for cholangiocytes, alpha SMA for smooth muscle cells, CD31 for endothelial cells, F480 for Kupffer cells, and CD45 marker for hematopoietic cells. So after co-immunostaining with EGFP and cell-specific markers, they confirmed that the EGFP expression was completely absent in cholangiocytes and vascular smooth muscle cells. You can see here. So these are cholangiocytes and the smooth muscle cells along the sides. But they were positive in case of the Kupffer cells, CD45 cells, and um, endothelial cells. And they confirmed that the hepatocytes were the main target cells for this mRNA to get transfected and to get translated and to express the protein. But few of the Kupffer cells, CD45 cells, and the endothelial cells were also showing the EGFP protein expression. After confirming that the hepatocytes were the main cells which were transfected by the mRNA, they evaluated the mRNA LNP, which is encoding for hepatocyte mitogen like hepatocyte growth factor for proliferation of the hepatocytes. So HGF is known to trigger the proliferation of hepatocytes and plays a key role during the liver regeneration, as I was explaining in the introduction. mRNA encoding HGF was injected in intravenous route, and the cell proliferation was assessed by the incorporation of 5-ethylene 2'-deoxyuridine EDU into the hepatocyte DNA. EDU is a thymidine analog which replaces the thymidine during the DNA proliferation. So whenever the DNA will get replicated, this EDU will go and bind in the position of the thymidine. And then we can stain for the EDU and confirm that whether the DNA is replicated or the cells have been proliferated or not. In this study, so for one group they gave poly-C RNA with lipid nanoparticle, and then the other group they gave HGF encoding mRNA at the dose rate of 10 microgram for 20 gram mice, and then they gave EDU injection intraperitoneal route at the dose rate of 50 microgram per gram of mice. And then after 5 hours, so they did the first sampling, and after 24 hours they did the second sampling, and then after 48 hours they collected the samples of liver. They checked the hepatocyte proliferation by staining for EDU. After staining, so initially at fifth hour time point, they didn't see any proliferation. Policy didn't show any proliferation, but there were few proliferation, and later they confirmed that they are CD45 cells by doing the co-immunostaining, which I'll discuss in the next slide. But HGF, there was no proliferation in the 5th hour time point. By 24-hour time point, you can see the proliferation, and the maximum proliferation was achieved by at 48-hour time point, and it confirms that the mRNA injected to the intravenous route is going into the hepatocyte. It is encoding with HGF, also express the HGF protein and will proliferate the hepatocytes. So to confirm, the policy RNA cells which were proliferated were not hepatocytes. They did the co-immunostimulation staining with CD45, and it found that policy mRNA caused proliferation were not the hepatocytes but CD45 cells, but the HGF with HNF4 alpha showed the hepatocytes, as well as they were positive for EDU as well as HNF4 alpha, which is a cell-specific marker for hepatocytes. And also they quantified the proliferated cells. Uh, uh, EDU positive cells were maximum in HGF injected mice than the poly C injected mice, and also EDU positive cell with HNF4 alpha were maximum in HGF injected mice, but zero in case of poly C injected mice. But the CD45 cells were more proliferated in poly C injected mice than the HGF, because HGF is going and transfected in the hepatocytes to cause the proliferation of the hepatocytes. After confirming that the hepatocyte mature genes like HGF will make the hepatocytes to get proliferated, they evaluated HGF and EGF encoding mRNA in different liver injury model like chronic liver injury model and acute liver injury model. First they did the chronic liver injury model. So they fed the CDE diet, as I explained in the introduction. CDE diet is choline deficient ethionine supplemented diet. This CDE diet induces steatohepatitis in mice, which is a symptom in non-alcoholic fatty liver disease. So they fed CDD diet, means choline deficient diet, for 1 week, and then they supplemented ethionine for supplemented the ethionine with the choline deficient diet for next 3 weeks, and then they gave the first dose of the injection, which was having the treatment groups were given with HGF mRNA and EGF mRNA, both in combination, also in individual. There were 5 groups, and also control group received poly C mRNA and luciferase mRNA. And once the means they stopped the diet before giving the injection. So this is not a persistent diet model, which I'll discuss in the next step. So they stopped the diet here. The CD diet was stopped after 4 weeks of feeding, and then they gave a second dose on 4th day, and then they collected the sample on T2 as well as T8 days for evaluating the efficiency of this mRNA in reducing the liver injury caused by the CDE diet. The parameters studied were lipid accumulation in hepatocytes, were studied by doing oil redwood staining on liver sections, and serum cholesterol levels were studied, and also serum alanine aminotransferase, that is ALT levels, were studied. Serum cholesterol levels were studied because once a lipid which is stored in the liver will get released into the bloodstream, so the cholesterol can be evaluated and get to know that if the serum cholesterol level is increased, the cholesterol has been released from the liver and liver is getting recovered. In that way, they evaluated the serum cholesterol. And serum ALT is a liver function enzyme. Means if the liver is getting cured or getting recovered, the serum ALT levels will come down. Oil redwood staining. So the first result. This is for poly C, is a control group, and this is a luciferase, this is also control group, and this is HGF and EGF mRNA injected group. There was so in this, so there is no reduction in this steatosis, but here we can see, still we can see the vacuoles in these groups, but here it is reduced uh, when we compare with on T2. They have not given the baseline picture. Um, so they found that the recovery of the liver on oil reduced when they injected the HGF and EGF combination mRNA, and also the cholesterol levels were increased on T8 with the combination of TGF and EGF. The lipids stored in the hepatocytes were released in the bloodstream. So they found increase in the cholesterol levels, and also in they evaluated the ALT levels. So the ALT levels were decreased, showing decreased to like the normal level, so that the liver is the recovering. The ALT levels were restored to normal in the treatment group. After doing the study, they did a persistent diet model, means they continued giving, because there were some values which were showing very similar to the treatment group. The control group showed very similar to the treatment group. So they started giving the dose. So they did this study with a persistent diet. They didn't stop the CD diet once they injected the mRNA. So they gave CDT diet for 1 week, and they supplemented the methionine with choline deficient diet for another 3 weeks, and then they gave the first injection of the mRNA encoding HGF and EGF, and the control group received the policy mRNA, and also the mice received the second dose of injection through intravenous route on fourth day. And they collected the samples on T2 and T8 for the evaluating the lipid accumulation in hepatocytes by doing oil residue staining, and also they evaluated serum cholesterol levels and serum ALT levels. And the results were like this. There was significant reduction in the lipid accumulation on liver sections by performing the oil residue staining. You can see each red dot depicts each hepatocytes accumulated with the lipids. The policy uh, policy is having more of red dots, means like more accumulation of the lipids when compared with the HGF and EGF encoding mRNA injected mouse group. This is T2 time point, and this is T8, means day 2 and day 8 time point. And there was increase in the serum cholesterol level when compared to the control group on day 8 time point, and also the serum ALT levels were decreased than the control group, than the policy group. This shows that the two doses of the HGF and EGF mRNA lipid nanoparticle will reduce the chronic liver damage which is induced by the CDE diet. And after confirming the efficiency of this mRNA treatment in chronic liver injury model, they did the same with acute liver injury model, which was induced by acetaminophen overdosing through intravenous route. First, so for this, to start with, all the mice used in this research were female mice, and estrogen levels in female mice will alter the liver injury which is induced by the acetaminophen. To avoid that, they synchronize the estrous cycle by inducing them with the return effect. Return effect means keeping the female mice in the male cage with a male bedding material where the male was residing for a few days, where its pheromones will be present. So they transfer the female mice in the male bedding material cage, and then they synchronize the estrous cycle. And after synchronizing, they fasted the mice for 14 hours uh, to make to get the consistency in the liver injury model. And then after fasting for 14 hours, they injected 550 mg per kg dose of APAP, that is acetaminophen. And after 24 hours, they injected the mRNA encoding for HGF and EGF, and also they injected policy mRNA, and then they collected the sample after 8 hours, means at 32 hours post APAP induction, and also 48 hours post APAP induction, with 2 time points to know the efficiency of this mRNA. And the parameters studied were the lipid accumulation in hepatocytes was studied with hematoxylin and eosin staining on liver sections, and serum ALT levels were studied, and also the tunnel positive cells. Tunnel positive tunnel marker is for the apoptotic and necrotic cells. If the cells are dying, they will be, we can use the tunnel marker to know that whether the cells are dying or not. The results were like this. After 32 hours and 42 hours time points in control group as well as the treatment group, uh, on 32-hour time point, still we can see the necrosis of the liver and altered normal architecture of the liver. In the control group, it's not cured because there is no treatment given in control group. Only the lipid nanoparticle was a common thing between the treatment and control group. But in case of the treatment group with the HGF and EGF encoding mRNA, on 48-hour time point, we can see the liver architecture getting normal with no necrosis and no steatosis. And also they evaluated the alanine aminotransferase levels. The alanine aminotransferase levels were reduced when compared, significantly reduced when compared with the control group, and they um, measured the tunnel positive cells. The tunnel positive cells were very minimal when compared to the control group, which shows that there were less necrotic cells in case of the treatment group than the control group. To summarize all the results, so they finally concluded that the nucleoside modified mRNA is an effective delivery tool to induce robust and timely controlled expression of hepatocyte mitogens and regenerative factors in a narrow time frame that is necessary to treat a features like non-alcoholic fatty liver, chronic injury, and also to accelerate the liver repair in acute acetaminophen-induced liver injury. And pros and cons of this paper are: they have used multiple liver-specific markers to avoid the false positive results, and also they have used multiple parameters in every experiment to come into conclusion, and also they have used multiple controls like polyC, luciferase as well as the EGFP in luciferase in some of the studies. And cons of this paper are the histopathology scoring data was not given, only the pictures were given, and also the reduced ALT level in control group in study number 3 has not discussed in the article, and they have used only female mice. In the future study, they can use both the sex to come into conclusion. And also future studies, we can use other hepatic mitogens and other in any, like we can use individual hepatic mitogens or in combination to come up with a more efficient treatment. And also we can use other very specific liver injury models like carbon tetrachloride liver injury model to evaluate the efficacy. And also we can do the long-term studies to know the effects or any um, side effects. And also we can use the mRNA therapy in other diseases and disorders. Thank you.
All right, thank you very much. I think what we'll do is we'll open it up for student questions first, and then if other faculty that are participating in this seminar have questions, we'll let them ask them after the students.
I had a question. So you mentioned the future study to look at the long-term effects of using this therapy. So is this something that's expected to be like, okay, we used it once and it worked, or is it something that you would continue using? I wasn't really sure on that.
Yeah, non-alcoholic fatty liver disease is a chronic syndrome, so this cannot be like cured with a snapshot because there will be complete altered metabolic uh, metabolic form of body. Further, to mitigate that, we have to give multiple treatment for this, multiple injections. But this is for transient expression. If we want to reduce the steatotic liver, to the normal form, we can use it. So some more studies are required to know about this, or to increase the dose, and those dependent studies and all have to be done.
Okay, thank you.
We'll open the questions up for everyone now. Dr. Adams has a question. I'll just let her ask it.
Good morning. Yes, I just had a question with regard to, was there any concern of this uptake or transfer of the mRNA to other organ tissues? It seemed like it honed primarily to the liver, but it also went to other organ systems.
Yes.
Would that be a concern, or did they look into that by chance?
Yes, yes. Actually, they have discussed in the discussion part, telling that like no any other health related issues were detected, and not all the cells were detected by looking at the study. So they have said that there were no any other side effects which were seen because of the transcription of this mRNA with other cells. Yes.
So they looked at, did they do histology or
Yes,
other?
yes.
Oh, so okay. It seems like it was a very thorough study. I mean, I think they really did a nice job in designing it. Great, thank you.
Thank you, Dr.
Did they use female mice for all of the mouse work or just that last study?
No, actually they have used female mice for all of the studies. And then I searched about this, like why they have used the female mice. Because female mice are more settings, when they compared with the male and female, the female mice are more susceptible for non-alcoholic liver disease models. So they have chose the female mice.
Do you recall who their funder, the funder was for this project? I assume it was probably NIH. And I guess I kind of wonder if that was the justification that they gave to NIH, because now for most NIH sponsored work, they require balanced gender in experimental animals unless you have a really good excuse not to. Right. Other questions? I think it's really interesting to see this mRNA as a potentially used as a therapeutic, in addition to obviously its usage to prepare vaccines for COVID and other infectious agents. So pretty neat.
So I had an additional question with regard to the dose. It seemed like they only had, I don't know if they discussed how they came up with the dose that they selected, but I was wondering, did previous studies sort of come up with this dose that they used throughout the study? And also, if there were higher doses, is there any concern for toxicity, safety issues and that kind of thing?
Yes, they have not discussed about the selecting the dose, but I have read few papers where the mRNA-based therapies, normally they have used 10 microgram to 15 microgram of the mRNA. In case of vaccines also, they have used that and they are finding like they are getting the results on them, but they have not discussed exactly like why they have chose the dose. Yeah.
So it was a reasonable dose based on
Yes,
what
yes.
you saw as far as the range.
Yes.
Yeah, I thought that was interesting. And then did they ever talk about toxicity, even at higher doses, or any kind of side effects?
No, they haven't discussed, so that has to be done in the next steps. Okay.
Great, thank you.
Do you know from your reading how many different mRNAs can be simultaneously used at once, or here they use the lipid nanoparticle as a delivery? I'm curious how many targets you could put in there.
As you know, I have read only one paper like this, like they have given in a combination. I was surprising like whether the coding, both the coding stands in the similar word, but it's not like that. The HGF encoding mRNA is separate and EGF encoding mRNA is separate, and both have been encapsulated in a lipid nanoparticle, and then they have injected. But in normally vaccine, I have seen only one protein getting uh, complexed in a lipid nanoparticle and getting injected or getting the responses.
Thank you.
I don't know. I will look into this one.
And I had one last question with regard to how this is relevant to your research. You might have mentioned this earlier, but can you use any of this technology for what you're doing in the lab?
Yes, currently I'm using the nucleoside modified mRNA to develop vaccines. So I'm synthesizing mRNA. So all the procedures like what I explained in the mRNA synthesis, I'm doing the same. But no animal studies yet. But we are in the process of developing the mRNA-based vaccine. But this is a therapy like which is an open field to start up with anything.
Oh, that's great. That's exciting. Are you thinking about using nanoparticles as well for delivery system?
Yes,
Yep.
yes, the same nanoparticle complex.
Ah, okay. Yeah. Well, this paper makes a lot of sense then. Thanks.
Thank you.
I have a hypothetical question for you. If you were to be author on this paper, let's say, and you were funded for one additional study to move forward from this study, what would be your next step? I know you had quite a few steps that you said need to be further researched, but if you only got to pick one or design one trial to move forward, what would be the logical next step?
I would like to do. I would like to find one more hepatic mitogen in combination to give with it, because the results at this time point where they have shown is not efficient. So I would like to make more efficient mRNA therapy by using any other hepatic mitogen singly or in combination with these mitogens. And then I will develop a study for that.
Thank you.
Thank you.
Any other questions, comments? Well, I think with that, we'll probably go ahead and call it for today. Definitely a very interesting topic and technology that has a lot of current real world application. And as you mentioned, kind of an exciting new frontier. So thank you very much for a very nice presentation.
Thank
And
you.
for the enrolled students, please remember to complete your peer evaluations, submit them to Canvas before Monday. All right, thank you very much.